So we've got minimum distance.
Oh, sorry. Would that be 0?
0.
Yeah. Sorry.
0 centimorgans, we've got 50 centimorgans. So what does it mean if two genes are 0 centimorgans apart? Physically. So, yeah. What's centimorgan a measurement of? What do we measure? What are we observing in a cell or in an organism when we're looking at genetic distance? What's going on inside a cell that we're actually taking measurements of when we calculate what does centimorgans mean? What's another measurement for genetic distance? Not centimorgans, not map units. What's the third unit that we talk about? Centimorgans, map units. What we calculate when we calculate genetic distance is first centimorgans, map units. Recombination fraction. So what are we measuring when we look at genetic distance? We're measuring recombination. We're measuring the amount of recombination. And 0 centimorgans equals 0 map units equals 0 recombination fraction. So anytime you calculate 0, that means that in meiosis, when crossover happens, you have genes A and B, say, they're so close to each other that all of these random recombination events that happen between sister chromatids during meiosis, one will happen, we've decided in class. We're going to simplify things and say one of those 4 red X's will happen every meiosis in every cell. So that if those two genes, A and B, are really close together physically, you will never, 0, observe recombination occur between them. So that would be a case where 2 of these chromosomes have capital A and capital B, and let's say this is a dihybrid. So you'd never see capital A with little b, because those chromosomes never break in between the A and the B, and you'd never see a lowercase a with a capital B. That's 0 recombinants. That's 0 centimorgans, map units, recombination fraction. So basically, you can't get closer than on top of each other. So minimum 0. So 50 centimorgans. What's 50 centimorgans in recombination fraction? It's not 50. It's related to 50.
It's a recombination.
It's a fraction.
Fraction, thank you.
So it's not going to be 50 because that's not a fraction. So what's, pardon?
It could be like 5 over 10.
Right. So what's 5 over 10?
Half.
0.5. Right. So 50 centimorgans is 0.5. So 50 map units equals 50 centimorgans. So centimorgans and map units are identical. You divide either of those by 100, you get the recombination fraction. So that's the fraction. What that really means is fraction of gametes with a recombinant haplotype. So let's look at the situation where we have 2 sets of chromosomes. So same genotype, dihybrid. Capital A over lowercase a, capital B over lowercase b. A semicolon because they're on 2 different chromosomes. Now, let's say that this was produced, that individual was produced by a cross that was originally this. So what are the parental haplotypes? From that parent and that parent. So they're gametes. Yes, AB. I need more information than that.
There's 4 of them, right?
Capital A, capital B.
Well, right, the one on the left only has capital A's and only has capital B's. So its gametes will only have a capital A and a capital B. And likewise, that other parent only produces lowercase a and lowercase b. So that's how you can get the combination of those two gametes, for example, can produce an individual of a dihybrid genotype. So what's the genetic distance between genes that are on separate chromosomes?
50.
That's the maximum, that's 50. So the question originally was, now 5 minutes have flown by, why is that the maximum? Well, if you want to look at the offspring of that cross, we'd set up a Punnett square. So how many gametes are going to have, if they're on separate chromosomes now, say for example we get a gamete that contains the capital A version of that chromosome. What fraction of gametes that have a capital A are going to have a capital B? There's two choices here. You can either have capital A with capital B, or you can get that chromosome with lowercase b. So what's the ratio?
104.
You have as many capital A, capital B's as you have capital A's with little b's. That's one of Mendel's laws. So you pick capital A chromosome, you've got 50% big B chromosomes, 50% of the little b chromosomes. So even chance, 50-50, that you get a capital A with a big B or a capital A with a little b. You could do this with a Punnett square. To generate your gamete haplotypes. So these aren't genotypes, these are haplotypes. So you have 4 different haplotypes. But the critical point here, when you have capital A with a capital B, is that a parental type or not? Where are the parental types up here?
Capital A, simple A. Oh, yeah, capital A.
These are the parental types down here. So that's a parental type. That's a parental type. So a capital A with a big B would be a parental type. And you make those gametes equally frequently as you make recombinant gametes. So there, just from that one example, we've produced 2 gametes. One's a parental type, one's a recombinant type. So what's the recombination fraction? What fraction of the gametes are recombinant?
2 out of 4.
2 out of 4. 1 out of 2, 0.5. 0.5, 50%. And that's why the maximum genetic distance is 50%. Because you always, if 2 genes are on 2 separate chromosomes, Mendel tells us half the time the combination of alleles is going to be the parental combination. The other half of the time it's going to be a recombinant combination. So 50%, 50 centimorgans is the expected maximum genetic distance. So that comes straight out of basically the biology of meiosis.
Yeah. Sweet. Well, thank you.
Yeah, you're welcome. Good question.
And do you record for the names for office hour?
Yes, I do. What is your name? What's everybody's name? And then I'll make sure that if you haven't... Let's take roll. I'll make sure if you haven't gotten your points yet that you do. Okay. Who's present that hasn't earned their points yet?
Alright, I'm Jeff Williams.
Yeah.
And do you need student ID as well?
No, that's fine. That'll be enough. Anybody else who hasn't?
Could be PR.
Yep. So have you been to an office hour and asked a question before?
No.
Okay. Are you going to ask a question today?
Yes.
Okay. What's your name?
Lynn.
All right. Last name?
Lee.
Okay. Then I will... Anybody else?
Monique.
Monique. Yeah.
MOUA. Brenda Perez.
Karina Perez. Yep.
Zero pounds.
Others.
Paula Yang.
Paula. Yep.
Sidney.
Woo.
Yeah.
Yep. Anybody? Anybody? I've seen.
Okay. I just had a question
Please.
about your X chromosome inactivation.
Yeah.
I was wondering if that could relate to humans as well, like as in like blotchy skin or
Okay, so X chromosome inactivation, relevance to humans.
yeah.
The only reason that X chromosome inactivation, we love to talk about the calico cats,
Thanks, Dr. Jones.
absolutely, is that their X chromosome contains a pigmentation gene. So if this was going to be applicable to humans, and I don't know if this is true or not, we would have to have a skin pigmentation gene on the X chromosome
Oh.
that would display differences whether one chromosome was turned off or the other. I don't know if that's true for humans. I doubt it's the case, or we would see probably more tortoiseshell humans. But yeah, any trait that's present on the X chromosome in humans, any gene that's present on the X chromosome that causes a trait would be susceptible to seeing that sort of mosaic phenotype.
Oh. Well, I just had a question because I know with Asians we have like Mongolian birthmarks
Mhm.
when we're born.
Mm-hmm.
So I was wondering why it's like it only happens to Asians. Well, like
Oh, so yeah, there could be, yeah, that's a reasonable hypothesis. Is it only females or is it
no,
males
it's
too?
both.
Okay, so then it's an unreasonable hypothesis. But yeah, that's an interesting question. But yeah, you'd expect if it was X chromosome inactivation, it would be limited to females. Others? We've got one empty chair if somebody would like to come in and sit down. Rapping about genetics.
I didn't really understand the concept of like DNA methylation and acetylation. So wondering if like you can go into that.
Sure. So, epigenetic marks. We've got DNA and we've got histones. So, both of these molecules, okay, there's our DNA molecule, and then we've got histones, which are these sets of 8 proteins that DNA wraps around, well, there you can see 7 in an octamer. There's 8 there, really, trust me. Right? DNA wraps around the histones, and that's what we call chromatin. And all of those histone proteins have both N and C terminal tails. So while the center part of the protein is well ordered, the protein adopts a particular conformation. The ends of the protein, the N-terminus, the amino terminus, and the carboxy terminus sort of hang out. And these are the 2 different molecules that get epigenetic modifications. So in DNA, yeah, there's all sorts of things to be concerned about in my office. I apologize. Don't injure yourself. In DNA, that molecule gets chemically modified by the addition of methyl groups onto cytosine. So the nucleotide cytosine gets a methyl group added to it. That's that methyltransferase enzyme that we were talking about briefly in class today. And the way that causes repression of translation or transcription is that it's the way it's been described to me, which I take at face value, I suppose, is that those methyl groups sticking off of the DNA sort of block some proteins from recognizing the DNA sequence they're trying to bind to. They're steric. They keep the proteins a little bit farther away from the double helix because there are these methyl groups sticking off. And so that prevents activation of transcription in eukaryotes, for example. So that's methylation as an epigenetic mark that causes a change in expression, reduction of expression usually. Histone molecules, or proteins rather, have lysines, right, one of the 20 amino acids. The tails of the histones, the termini, tend to have an abundance of lysines. So there are lots of K as the single letter code for lysine. There are lots of lysines in their tails. And those lysines can be methylated or acetylated. Different enzymes. Histone methyltransferases and histone acetylases, histone acetyltransferases. You don't need to know the names of the enzymes. And the point there is that while DNA methylation sort of directly affects transcription sterically by blocking things from coming and interacting with the DNA, histone tail modifications, acetylation, methylation, are some of many, but they're the most important two, I think, work by changing chromatin structure. So acetylation leads to, I've put myself on the spot, I don't remember. Acetylation, if I remember correctly, leads to euchromatin, open chromatin structure, where the histones are far apart. DNA is still wrapped around them. But they're farther apart, the DNA is loosely associated with the histones. And methylation, if I'm remembering correctly, leads to compact chromatin structure, where you get the nucleosomes bunched up right next to each other, so there's not much room for other proteins to come in and turn on transcription to transcribe genes. So that's the difference between the similar modifications, including methylation, happens on both sides of the figure, but it works in different ways. So when it's methylation of histones, that's changing the structure of the DNA molecule itself, the chromatin structure. Direct methylation of DNA has a separate effect, like we talked about first.
So I have
Yeah,
a question about
please.
the cis and trans regulation. Could you go more over this diagram?
Okay, so cis and trans. So these are both types of regulation. And I brought it up in the context of, was that the lac operon? Let me make it even, so I could talk about it in the context of that figure. Let me make it a little bit more broad, because this is not a concept that's specific to lac operon regulation. So there are two different ways to regulate, regulation of gene expression that we've talked about so far, lac operon and GAL utilization, relies on two different things. We've got DNA, obviously, because that's the thing we're trying to transcribe. So we have to have DNA in the picture somewhere. And then we've got the things that actually do gene expression, the proteins, the enzymes. So we have DNA, and we have proteins. So if you've got a promoter, which is the region of the DNA that has sequences that those proteins will bind to and recognize. If it's a repressor, that means it'll turn off transcription. If it's an activator, it'll turn on transcription. Those proteins are encoded by a gene somewhere else, like way over here. There's a transcription unit, a gene that produces that protein. If I mutate the promoter so that this protein, or any protein, doesn't recognize the sequence, let's say this is a repressor. Let's say this is a repressor of the sequence ATAG. It'll bind to that 4-nucleotide sequence and it won't let go, like lac repressor. Let's say we have a mutation in the promoter. There's usually an ATAG there, but now it's been mutated, that 3rd nucleotide, to T. So now it's ATTG. And let me make this a little bit more complicated, although not complicated, by saying there's another gene down here that also has the same recognition sequence, ATAG. Sorry, it's upside down for you, but I'm running out of room. Now, if I make this mutation to this promoter, but not to this promoter, let me number these genes, or name them somehow. So that's gene A, this is gene B. All right? So we are going to look at gene expression of both of these genes. They're both regulated by the red repressor protein. So if I mutate this promoter and not this promoter, what's this protein going to do in this cell?
Or bind to the B.
It binds here just fine. It recognizes that sequence. There's the repressor. What happens up here with this mutant promoter?
Doesn't bind to it.
Yeah, it doesn't bind to it. So we don't see that. No binding. So that means that in this circumstance we get production of protein B, we get no production of protein A. That makes this mutation have an effect in cis. What cis means is together or next to each other. This is totally different than cis in organic chemistry, by the way, so hopefully that's unfortunate. Yeah, we got ortho, meta, para, cis, trans. In this case, cis does not mean on the same side of, or anything like it does when you're looking at double bonds in organic chemistry. What this means is things that are cis, or effects that are local or nearby. So the critical point here is that that mutation only affects the expression of genes it's near, cis to. I contrast that then with trans. So where's a mutation in this drawing that I can make that will have a trans effect on expression of both genes A and B at the same time? I'll say what everybody was thinking. So what if we made a point mutation in the gene that encodes the repressor that eliminates its DNA binding domain? So it can never, regardless of the DNA sequence, it can no longer ever bind DNA and repress. That means that what happens to gene expression for gene A and gene B? We don't have a repressor anymore. So gene A will get transcribed. Gene B also doesn't have a repressor that can bind to ATAG, so it gets transcribed. So now we have a trans effect. One mutation over here is influencing genes that are far away, not just regulatory sequences like in this case, the promoter right next to this gene. If you mess with the promoter, it only affects expression of this gene. But if you have a mutation that has a widespread or trans effect, that's the difference between cis and trans. Cis is a local effect. Trans is more of a global effect. The easiest way to summarize this, I think, and since what I think matters in this case, I'm just going to put it out there, is that cis mutations are almost always regulatory mutations to regulatory DNA sequences, because those sequences act locally to regulate the gene that they're next to. Trans effects are almost always, they're trans, they can act over long differences, usually because they're diffusible, because you've created a mutation that affects a protein. So that protein, it's encoded way up here, but once you produce that protein, it can go to any molecule of DNA anywhere in the nucleus and have an effect. So that mutation that's way far away, wherever that protein is encoded, has effects, can have effects at multiple spots on lots of different chromosomes all throughout the nucleus. That's the trans concept. So, to try to wrap it all up, the point of that lac operon table that I handed out was that you can't if you have a cis regulatory mutation, you can't usually fix that by adding a wild type copy. Usually. This is not a blanket, this is a 99% statement, not a 100% statement. Because if it's a regulatory sequence, like we drew, here's the operator, here's the transcription start site. If I inject a plasmid that has a wild type operator, so let's say that's a mutant operator sequence, here's a wild type operator, O+. That doesn't help the situation because the operator in this diagram that's controlling expression of this gene is this one, not this one. So it doesn't matter if this is wild type because that doesn't help this transcription start site get used for transcription. It's on another piece of DNA far away. But if you have something like a wild type operator and you've got, what's the gene for lac repressor? It's LAC-I, I think. If you've got a mutation to LAC-I, like, I don't know, which one was the, super repressor. Let's say it's the super repressor mutation, LAC-IS, where it always binds the operator and never lets go. If we provide now a wild type copy of that, I+ on a plasmid, you're going to have mutant repressor and you're going to have wild type repressor in the same cell. So both of those proteins have access now to this wild type operator, and those are trans effects. So you can put this wild type version of a protein in a cell, that protein gets produced, it can have effects all globally, all trans, all over the place. But when you put a wild type copy of a regulatory sequence in a cell, that doesn't affect what's going on up here. So those are cis effects. Really beat that horse to death. So, yeah.
So somewhat related to that, I was just kind of looking over the notes, and I was wondering what different lac repressor genes were and the effects that they had on methylation. I saw there was a LAC-I and then the LAC-O.
Okay. Yep. Different tool. LAC. Okay, we've got, what were the different mutations we talked about? We've got LAC-O is, or what are the different genes that are involved? There's LAC-O, there's LAC-ZYA, there's LAC-I. So O is the operator. That's a DNA sequence. LAC-I encodes the repressor. That's the protein that recognizes and binds to the operator. So the operator is not a gene. It's DNA, yes, but it doesn't encode a protein. And then you've got 3 genes that are right next to each other that get transcribed together, Z, Y, and A. So the operator is a specific DNA sequence up there, and somewhere else on the chromosome is LAC-I, the gene that encodes the repressor. So that's the whole set of sequences that we've talked about. 1 encodes the repressor, 3, Z, Y, and A, we haven't talked about what they do, but they're involved in the bacteria, or the yeast bacterium, actually taking up lactose from their media and metabolizing it. So they're enzymes, basically. Other questions about the lactoperon?
So when you're doing a transcription of a LAC-O, how exactly would that look?
So because LAC-O is not, LAC-O, the operator, isn't a gene, it doesn't, that's okay, it doesn't get transcribed.
Okay.
All that happens to LAC-O is, well, it's in the promoter. So what happens with promoters? What's going to bind to the operator? LAC-I. the protein, which is lac repressor. So unfortunately, we've got two names for things. Not my fault. I'm just passing on the bad news to you. So LAC-I, the gene, gets transcribed into messenger RNA, and that messenger RNA gets translated, and that's what produces lac repressor, the protein that binds to the operator sequence. And when repressor is bound to the operator, no transcription of lac Z, Y, or A happens. So we have to, if we want to produce Z, Y, and A proteins, these fancy little blocky blobs, we have to get rid of the repressor from the operator. So this protein needs to unbind in order for RNA polymerase to come in and transcribe lac Z, lac Y, and lac A.
So I have a related question.
Mhm.
Can you go over the OC mutation, I minus IIS mutation, and how lac repressor mutant works?
Okay. So if it's an I mutation, where does that mutation occur? Where is that nucleotide change? It's a trick question. It's the obvious. In what gene or what part of the DNA molecule? If it's a mutation that's got the letter I in it, where's the mutation? In the genome. The whole chromosome of bacteria. If it's a LAC-O mutation, where's the mutation?
Operator?
Yeah. So we've got the operator. We've got lac Z, lac Y, lac A. Somewhere else in the chromosome, we've got the gene for lac-I. So if it's an I mutation, it's a mutation to the lac-I gene. If it's an O mutation, it's a mutation to the operator. So let's start with the OC mutation. So if you make a OC mutation, that changes the operator sequence. So what's the effect of that?
Always on.
Constitutive. So why is transcription always on in this case? What is the effect? What's the immediate effect of mutating the operator sequence? What doesn't happen anymore? What binds lac operator? The repressor. It's recognizing a particular DNA sequence. The operator, OC is not just a single mutation. There are a number of different specific mutations that can cause the same effect. You change the sequence of the operator, so lac repressor never binds. So if your repressor never represses, this is double negative, no repression equals constitutive, always on. Transcription, 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 all the time, unregulated. Because the protein repressor, lac-I, doesn't recognize the new operator sequence. So that's the OC mutation. That's the only one we've talked about where there's a mutation at the operator itself. The other two are mutations to lac-I, the repressor protein, or that change the protein.
So it doesn't repress anyone.
Well, one of these is a, what's S? in LAC-IS, S stands for what? It's my horrible attempt at making a
Super?
Superman. Yeah, super repressor. What sort of mutation could you make to LAC-I, the repressor, that makes it a super repressor? So it represses by binding the operator. In what circumstance does LAC repressor let go of the operator and let transcription happen? When is it good for the bacterium to have transcription of the LAC operon? What sort of an environment? When do we want transcription of LAC-Z, LAC-Y, and LAC-A? What's the purpose of the LAC operon? What's LAC? Why LAC? Why is it called LAC? Something about a small molecule, something about lactose. Right, so when the bacterium has lactose as a sugar source, it needs to produce the proteins LAC-Z, LAC-Y, and LAC-A to use it to grow. So we want transcription when lactose is present. We turn off transcription by using LAC repressor. So when LAC repressor binds the operator, there's no transcription. So in the presence of lactose, we want to get rid of LAC repressor. How does lactose get rid of LAC repressor? So something happens. When lactose, that small sugar, is available, LAC repressor leaves the operator so that transcription can happen. What's the interaction between that small molecule lactose and LAC repressor? What happens? So lactose physically binds LAC repressor. There's repressor, there's lactose. So what happens when lactose goes and binds into its site on LAC repressor? What sort of a site is this that it binds? Talked about it in class today. Yeah, allosteric site. So lactose binds the allosteric site on LAC repressor, causes LAC repressor to change its shape, and because it changes its shape when it's bound to that small molecule, it no longer temporarily, just because of its shape, no longer has a DNA binding, not domain, surface. So the protein is no longer in the right shape to recognize and bind to the operator. And that's how lactose gets rid of LAC repressor so that transcription of the LAC operon can happen. What could we do to LAC repressor, getting back to the IS mutation? That's normal. That's the usual situation in bacteria, how this works. What could we do to LAC repressor to make a super repressor that always represses and never can be gotten rid of?
Change the shape of where it binds.
Binds DNA or binds lactose?
Lactose.
Perfect. So that's a mutation to the allosteric site. Now note, that's a mutation I'm sure you understand this, but that's a mutation up here in the DNA that encodes the protein. But it causes the production of a protein that, say, doesn't have that allosteric site. So it gets rid of the allosteric site, so it can't be regulated by lactose anymore. So presence of lactose, absence of lactose, doesn't matter. All lac repressor is going to do is what it knows how to do. It's going to go bind DNA and repress, and you can never get rid of it.
So this causes lactose intolerance.
No, had nothing to do with lactose intolerance. Well, first, this is bacteria, not humans, but we can talk about that more. It does, but in general, no. Okay, so last one, I minus. So I minus is another constitutive mutation. And now I've drawn in my I minus spot in the table. So like the OC mutation, lac I minus is a constitutive mutant. Transcription always happens. So it's not a mutation to the allosteric site of lac I now. It is a lac I mutation. So what else can we change about lac repressor, lac I, that affects its ability to do its job? So in this case, if transcription is always on, what's the logic here? We want to make a repressor that can't repress.
So you use a repressor to repress the...
It's a mutation to this protein, though. So it's not another protein.
Oh.
It's a lac I, a mutation to this protein. So we have to do something to this gene that encodes this protein that makes it never able to repress. So what's the essential function? Clear your minds. Close your eyes. Imagine you're somewhere else. What's the essential function of the lac repressor molecule that makes it a repressor? What does it have to be able to do to repress transcription?
It should bind to the operator to stop.
It has to be able to bind DNA.
Mm-hmm.
That's how this protein represses. Is it sits there on the operator and it physically blocks RNA polymerase and other proteins from coming and trying to transcribe. So its key function is binding DNA. So what happens to lac repressor if we mutate instead of IS, where we change the allosteric site? What happens if we make a mutation I minus that produces a protein that has no DNA binding domain? So that's what the I minus mutation does. It gets rid of its ability altogether to bind DNA. So a lac repressor never binds the operator, never represses. Transcription's always on. It's yet another way to get constitutive expression. Because it's a two-factor system, repression rather. You have to have two things. You have to have the operator and you have to have which is the DNA sequence. You have to have the protein, lac repressor. They have to work together. So if you mutate one, the operator, or if you mutate the other one, the repressor, you have the same effect. No binding. You mutate the operator so that the repressor doesn't recognize it, you get constitutive expression. Or you mutate the repressor protein so it doesn't recognize the operator sequence, they don't interact, you get constitutive expression. That's all I know about the lac operator system.
So is this something similar in humans? Something similar that happens in humans?
Well, so lactose intolerance does have to do with enzymes, turning them on or off, enzymes that are used to digest the sugar lactose. But we don't have lac repressor. So there's a whole different set of genes that regulate whether or not transcription happens.
Thank you, Doctor.
Absolutely. Have a good afternoon.
Cannot be this lactose intolerant, can't it be epigenetics?
Mm-hmm.
Because if a
Sure.
bunch of people that never use dairy milk for like a lot of generations and they developed the thing.
Yep.
So the body didn't
Sure.
need that enzyme.
Yep. Yeah. So it could be genetic. It could potentially be epigenetic. Yeah. Good. Other questions? Oh, you just missed it.
The cis-trans question? Oh, we did?
I recorded it though, so if you'd be willing to
Okay,
watch,
okay,
I'm going to
yeah.
post this later. So if
Okay.
you're willing to, and then I will certainly answer questions, but I'd like you to watch it first.
Okay. And I had a question,
Yeah.
on last week when you did the chart regarding the FOXP3
Yeah.
and the Fresno non-asthma, Fresno asthma.
Oh yeah, yeah.
When drawing the chart, does, I didn't know if the graph going upward meant more heavily methylated or if it was less methylated.
Okay.
When,
So FOXP3 methylation should be more methylation is higher.
okay.
So
Oh, okay. Because I did it backwards. I see now. So therefore, it should be a downward slope.
let's see. Yeah, and I think I stuck in the actual data from the paper, the figure that showed the actual plots from the research. But yes, the idea was that Fresno individuals with asthma had a lot of methylation of the FOXP3 gene. And there, by the way, I didn't bother correcting this in class, or at least mentioning it. There's no reason to expect a negative slope, because what's the x-axis? It's cities and
Very
disease
city-
states.
Yeah.
If you put Stanford over here, you have data going all over the place. So the trend was not important. It's just understanding how methylation should interact with
Okay.
the disease.
So on a chart like that, usually it's more methylated on the is upward.
Yeah.
It's not like the FOXP3 gene is more activated than.
So it really just depends on whatever the y-axis label
Okay.
is. So yeah, if it's FOXP3 activity, then
It can be reversed.
so methylation up would mean low activity. So if it's FOXP3 activity, then you'd expect low in Fresno with asthma, and then higher, and then higher, and then higher. In the way that the authors drew the x-axis. Yeah. So yeah, it's important to be able to be comfortable enough to play with, is it methylation, which is the opposite of activity in epigenetics.
Mm-hmm.
When DNA is methylated, that usually means there's less gene activity, less gene expression anyway. So they're oppositely related to each other.
I just had a question about system trans, but I'll just look at the video.
Please do. Yeah, I appreciate it. I know I talked about it in class once, so maybe watching a video doesn't help, but I explained it a different way. So see if that helps.
Okay.
And email questions, call me, ask me questions after class, next office hours, in review session if you want. Because I'm sure a lot of people have been asking me about system trans.
Yeah.
Not everybody yet. So I know that it's a
Once-
question
once
that a
they
lot
get
of people
there, yeah.
have. Yeah. Yep. But here, the basic summary of cis and trans is that cis regulation usually has to do with DNA sequences. Trans usually has to do with proteins. So cis means next to each other or together.
Yeah.
And that's usually because the regulatory sequence that controls the gene is physically next to it. So if you mutate the promoter, that only has effects on the gene that it's next to, cis effects. Whereas if you make a mutation that affects a protein, that protein then can go and operate at multiple genes throughout the nucleus. And so it has effects not just related to the original mutation nearby on the DNA, on the chromosome, but that mutation, because it's to a protein, then that protein can go interact with lots of other. So it has a broader effect or a trans effect. It's a good rule of thumb. Almost always true.
So crossover trans is different because it's not actually next to it. So
Right.
it wouldn't really affect it.
A transmutation will have a broad effect because you've changed in the usual circumstance in which cis and trans is discussed. Transmutation, mutation to a protein, then that protein can go have its mutant effect, if it's a transcription factor, for example, on all of the genes throughout the throughout the genome that it regulates. But a cis effect, a mutation of a single promoter, will only affect that one gene that the promoter is controlling.
Okay.
So it's a limited effect, local.
Okay, that makes more sense.
Yeah. Now, did you want to ask questions about the presentation, or you want to actually do the presentation? Do the presentation would be better as a video. You can give it to me in person, but I'd rather, it needs to be to the class. So that's why I was asking people to record it and put it on the box as a video.
I can record it.
Yeah, if you want.
Yeah.
Got 13 minutes left, so.
Okay.
Yeah, if you want to record it while you're telling me about it, that's fine.
Yeah.
Other questions? Stick around and listen to a presentation. Are we doing comedy, or are we doing?
It's about the C. elegans and epigenetics.
Oh, okay, cool.
Actually, I have a quick question.
Yeah, please.
For the test,
Yes.
can I request that you grade my test instead of Fatima? Or not?
No.
No?
Nope. Everybody's test gets graded the same way. That way it's consistent. Yep. You can request it, but no.
No? Okay.
Alright, thank
Yeah.
you, Dr. Russell.
Have a good afternoon, gentlemen.
I can plug my laptop into this thing.
Yeah.
But then,
And I also had a question about like the office hour points. So how does that work? Are we gonna?
If you tell me that you were here today and asked a question, then I will record that you were here and I will give you points.
Okay, yeah. Hi, Athanasian.
And
Anthony Ayerza.
right. Did you see my comment on your video?
Yeah,
I like-
we did.
yeah.
Yeah, thank you.
Nice job.
Thank you.
Appreciate it.
Alright, goodbye.
Yep.
thank you.
Okay.
I don't think I will share my book.
Oh, you don't need to. You can just show me. Your.
Oh.
Yeah. I don't need to plug it up there.
Oh.
That's How's life?
It's alright.
Yeah.
I wanted to ask if you had got my first extra credit, because I submitted 2 with both lectures. But we hadn't gotten feedback from presentations. But I was hearing that you
Well-
said that people got 2 or 3 or not, so I'm curious. Since I didn't hear back, I assume you didn't get it, because
Well,
I didn't points for it.
That's the only feedback I gave.
Okay.
Either points on Blackboard or not. So, just a second. Let me check.
Because I wouldn't have mentioned it, but I was like, it looks kind of strange.
Basically-
People kept saying stuff.
Basically, if it's not there now, then I didn't receive anything from you.
Okay.
So, just a second. Yeah. Check. Yeah, your name wasn't on the sign-in sheet, and I didn't get any submission from you.
Okay, but what about the second one with the GMO?
Yeah.
Okay.
Yeah, you got 2 points on Blackboard for that.
Okay, because I know the photo didn't show up, so I was like, wow.
That's probably why.
Yeah.
So yes, I didn't get anything from you for the first extra credit, but for the GMO one, yes.
And then the comedic genius, you said you had to make your own joke, correct?
You've already got full points for that, so you're good.
Okay. had the joke already out, so I was like, do I just tell it to you?
You can if you want to, but you already got the points for that category of points. Comedy it's comedy and current genetics together
Oh, okay.
for 7 points total. So you already got 7 points.
Okay.
You don't have to worry about it. But you can tell me a joke if you want.
Sure. It's kind of long though. Is that okay?
Yeah?
Okay. So, a boy got a large gash on his arm, and his sister, type AB, quickly gave him a transfusion from her blood to save him. Instead of feeling better, he started groaning loudly. His sister, out of concern, goes, "Oh my gosh, are you okay? What's the matter?" And he goes, "Ugh, meiosis."
That works.
Yep.
I like it. Kind of, you got the immunology, you've got the genetics.
I thought it was cheeky. If we had questions, would it be better to come before Wednesday? Because I know a lot of people are probably going to be showing up at your office in the morning.
So we've got, we've got the hour before class, and then we've got the hour of review during class.
Yeah.
So
is it better to just review during class? Or, because I know you have a lot to go over for review.
I'll just answer questions until people stop asking or until we run out of time. So, if you want to get in class and get your hand up early and get your question answered, then that's fine.
Because
Yeah.
I was going over the exams, and it seems I have more questions than I thought I would. So I was kind of concerned.
Well, hopefully they all get addressed, but if not, then let me know and we can work on what to do to get those questions addressed.
Okay. Thank you.
Yeah, absolutely.
Good topic. Sorry. I didn't mean to look.
Bye.
Take care. Let's see.
Just figuring out a way how to record this thing.
Well, let's not be too concerned. I can record it actually
Okay.
if you want.